All right, let's talk about glycolysis first. Glycolysis takes place in the cytoplasm and does not use the mitochondria or oxygen. Glycolysis makes a small amount of ATP, and it also generates some high energy electrons that are going to be carried by NADH into the mitochondria for later use. Now, as far as number of carbons here, glucose has 6 carbons. Each one has at least one bond to an oxygen already. Glucose is going to be split into 2 pyruvates, and you'll see the structure of that in a minute on another slide. So if we look at the word here, glyco is sugar, lysis means split, so glycolysis is just literally sugar splitting. We're going to split glucose in half, and because we are reducing NAD plus to NADH, that means that this is an oxidation reduction reaction. Then we'll talk about the next step here. Pyruvate is going to be transported into the mitochondria and it's going to be oxidized. That's why it's called pyruvate oxidation. And we're going to go from a 3-carbon molecule to a 2-carbon one. So acetyl in chemistry just means 2 carbons. So Acetyl-Coa Coenzyme A is a large molecule. It's one of those molecules that's an organic molecule that doesn't fit into one of the 4 categories of molecules that we studied. It's not a carbohydrate, a lipid, a nucleic acid, or a protein. It doesn't quite fit into any of those categories, but it's used to move around this 2-carbon bit here. Now, after glycolysis, most of the energy is still in the pyruvate. And even after pyruvate oxidation, most of the energy is still in that 2-carbon acetyl group. Now, then we'll talk about the next step here, the citric acid cycle, which is also called the Kreb cycle, Krebs. The remainder of the energy that's found in this carbon backbone is going to be removed, and carbon dioxide is going to be released in this step, and we'll go into the details of this in a minute. The remainder of that energy is going to be carried away in high energy electrons, NADH in its reduced form here, and also we see a second electron carrier here, FAD, which when it's reduced is FADH2. Now FAD is needed because it can grab electrons that are a little bit lower in energy than the ones that NAD can grab. So that'll be an important part of this. So we generate a little bit more ATP here from this step, and then we'll talk about the last step here. Here's where the electron transport chain is. All of these high energy electrons, more electrons, they're all going to go to the electron transport chain. And there's a lot of words here that are confusing. So oxidative phosphorylation. Oxidative just means we're going to use oxygen. Phosphorylation, that word just means we're going to stick a phosphate on something. So if something gets phosphorylated, that means a phosphate was stuck on it. And remember, a phosphate is just a phosphorus with 3 or 4 oxygens bounded to it. So oxidative phosphorylation is going to involve 2 steps. The electron transport chain, that's the oxidative part, that's going to use oxygen, and chemiosmosis. This sounds confusing, but this is going to be a hydrogen ion gradient that's going to flow across a membrane. So it's not osmosis, it's chemiosmosis, and this is going to be used to phosphorylate ADP to ATP. So that's where the phosphorylation part comes in. So there's a little movie in here that's from our textbook manufacturer, and you can find this to watch it in the link in the PowerPoint. I'm also going to try to find it on YouTube and put it in this playlist because I believe it's on YouTube as well, because it's not brand new. So I'm not going to show it to you right here. The process that generates most of the ATP during respiration is that last step, oxidative phosphorylation. Oxidative phosphorylation is about 90% of the ATP that's generated. That's why when we don't have oxygen, ATP production quickly falls off, because that's where most of it is coming from. Now, if you look at an older source or a source that's a little bit out of date, you might see a number of 38 molecules of ATP per every glucose molecule. And that 38 ATP per glucose, they got that just by counting all the electrons that are generated in these ATPs and then figuring out exactly how many hydrogen ions were going to flow across the membrane and how many. So that's kind of the theoretical maximum. Well, now that we can measure ATP production in cells directly, although it's pretty difficult to do, we know that no cells get close to that. So the real maximum is about 32 molecules of ATP per one glucose molecule that was there at the beginning. So human cells are pretty inefficient and don't even get to the 32. But they're still, it's still generating a lot of ATP, even though a lot of those high energy electrons end up being lost and they are not used for making ATP. Okay, so back to glycolysis. Let's look at the details here. So the overall reaction, and this is something that's going to be important to know for each step of this process, what goes in and what comes out of each step. So for glycolysis, glucose goes in, and what comes out is 2 pyruvates, 2 net ATPs, because we have to use ATP in order to destabilize the molecules and break things up. So we're going to harvest 2 ATP net because we have to use some ATP to get things going here at the beginning, and some high energy electrons. So that's what comes out. 2 net ATP, 2 pyruvate, 2 reduced NADH with their high energy electrons are going to be produced as part of this process. So I have taken out of this diagram the names of the enzymes because you don't need to know those, but each reaction here, each blue circle, that's a different reaction that's catalyzed by a different enzyme. So here's the first 5 reactions here, each with a different enzyme. And I've taken out as well the names of the intermediates because you don't need to know those except for this one. Glyceraldehyde 3-phosphate, which is abbreviated as G3P. G3P is an important metabolic intermediate. G3P is where things can come in and out of this reaction. Glycerol, as you might remember, is a 3-carbon sugar. That's the backbone of triglycerides. 3 fatty acids are attached to a glycerol to make a triglyceride. So if triglycerides are being broken down, glycerol can enter this reaction here. Glyceraldehyde can also leave the reaction here and go off to make amino acids, to go off to synthesize triglycerides and be the glycerol backbone. To be used to synthesize phospholipids. Glycerol is also part of the structure of a phospholipid. Okay, so G3P is an important intermediate. That's why I've left that one in here. So here's, we're using ATP here to destabilize the glucose molecule so that it can be split. So here is an oxidation reduction reaction. And I know this is an oxidation reduction reaction because NAD is being reduced to NADH. So I'm going to go back a slide here again. So if we look at this molecule, G3P, because we know it's going to get oxidized, so look at the bonds here. So we have this carbon has 1 bond to oxygen, this one has 1, this one has 2. So we know something is getting oxidized here because something got reduced. So this carbon here now has 3 bonds to oxygen instead of 2. So this carbon is the one that got oxidized here, NAD plus got reduced, because it's always both. If something is obviously being reduced, something else got oxidized. Even if we don't notice it right away or we're not sure what it is, something else had to get oxidized if something got reduced. Okay, so now we keep going. Here's pyruvate way down here. So there's a total of 10 enzymatically catalyzed reactions in glycolysis. This process that we think of as kind of a simple part at the beginning of cellular respiration, of just splitting glucose into 2 pyruvates, it's actually quite a complicated process that uses 10 different enzymes. So it generates 2 net ATP, because we have to account for the ATP that we used at the beginning, the 2 ATP that were already used. So we get 2 net ATPs, 2 pyruvates, and 2 NADH with their high energy electrons. So that's our outputs. So for each of these reactions, that's an important thing to know, is what goes into this particular reaction and what comes out of that particular reaction. So the next step here, pyruvate oxidation. Pyruvate enters the mitochondria and is oxidized. So that means something else is going to be reduced. If anything gets oxidized, something else is going to be reduced. So here's the structure of pyruvate. Now, if you notice here, this carbon is already almost carbon dioxide. That's what's going to be oxidized. So this carbon here is going to become fully oxidized to carbon dioxide. There's our first carbon dioxide that's released. And you notice we haven't even used oxygen yet. A common misunderstanding of cellular respiration is people say oxygen is turned into carbon dioxide, and that is not true at all. They're used in completely different steps. So here we have pyruvate oxidation. We're already getting a carbon dioxide being released, and we haven't even consumed our oxygen gas yet. Okay, so this is an oxidation reduction reaction. So what's getting reduced here is, again, NAD+. So pyruvate oxidation is going to generate a couple more high energy electrons, a CO2 for each pyruvate, and our end product here is acetyl-CoA. So here's our acetyl group, our 2 carbon acetyl bit. There's still a lot of stored energy in this molecule. We have a carbon-carbon bond here, and this carbon has 3 bonds to hydrogen. So that's still quite a bit of stored energy left in that molecule. So now this is inside the matrix of the mitochondria. So you notice here they're showing 2 membranes here, and that inner membrane, that's the membrane of the ancient bacteria. So of course, inside in the cytoplasm of the ancient bacteria, which we call the matrix of the mitochondrion, that's where the next reactions are going to take place, and that's where these reactions are taking place as well. And you'll notice how we write CoA here, capital C, lowercase o, capital A, because Co is short for coenzyme. So coenzyme A. Next is the citric acid cycle.